Thank you so much for this nice introduction, and I'm really excited of being here. As you mentioned, I'm Bianca Graziano, and I am a postdoc in the lab of Dr. Helen Wilsey at UCSF. And today I'm going to talk about my research. I'm going to talk about the role of STXBP1 beyond the synapses, focusing in particular on its role at the cilium. I think here we all know that traditionally STXBP1-related disorders have been considered synaptopathies. We know that STXBP1 is a fundamental regulator of the SNARE protein complex, and that this SNARE protein complex mediates vesicle fusion at the presynaptic terminal. And we know that when this process is impaired, this leads to the development of neurological symptoms, epileptic encephalopathy in patients. But we also know that on top of the neurological symptoms, patients can also develop pulmonary comorbidities, which accounts for up to 1/3 of premature deaths in patients. And conventional wisdom tells us that these pulmonary complications are associated to the neurological symptoms. They are caused by the hypotonia in these patients and by the role of STXBP1 in these peripheral neurons in the lungs. But it is also well established that in the airways epithelia, in the pulmonology field, in the airways epithelia, there are these highly polarized cells. For example, there are globate cells which produce mucus. There are ciliated cells whose ciliary beating is really important for redistributing this mucus and creating a mucosal barrier that protects from pathogens. Whenever their cell function is disrupted, this can lead to the increased number of infections and a prolonged duration of infections. Importantly, these highly polarized cells heavily rely on targeted vesicle trafficking for their function. And if we zoom in on a multiciliated cell type and we look at the microscopic structure of a single cilium, we can see that at the base of the cilium, there is actually a diffusion barrier that needs to be crossed by vesicle trafficking in order to deliver proteins and cargos to the ciliary compartment. So considering what we know about STXBP1 and its involvement in this vesicle trafficking, we wonder whether STXBP1 might also have a role at the base of the cilia for this type of process, and therefore we hypothesize that also ciliary STXBP1 might be contributing to the pulmonary comorbidities on top, of course, of the role of STXBP1 in the peripheral neurons. Since this was kind of an unheard concept, we decided to check if STXBP1 was present in this type of cell at all. And to do so, we looked at a previously published transcriptomic data set obtained from developing human airways. And we subsetted this data set into a peripheral neuronal cluster, which is associated with the neuronal marker MAPT, and into a multiciliated cell cluster. And of course, we found that STXBP1 was expressed in neurons, so we had no doubts about that. But we also found that STXBP1 was expressed in multiciliated cells. Since STXBP1 interacts with SNARE proteins, we also wondered whether SNARE proteins would be expressed here. And we first looked at the canonical synaptic SNARE, syntaxin 1, SNAP25, but we found very low expression. Fortunately, previous work from the State Lab in which they looked at a ciliated human cell line, they found that they performed mass spectrometry and they found that it expressed STXBP1 and that STXBP1 interacted with syntaxin 2 and syntaxin 3. So following this line, we looked for these non-canonical syntaxins, and we found that these non-canonical SNARE proteins were also expressed in the multiciliated cells, which hinted to the fact that maybe STXBP1 is interacting with different binding partners in the multiciliated cells as compared to the synapses. At this point, we wanted to perform a localization experiment, so we turned to our frog model. This is a really well-established model organism. And in addition, it has very important similarities with the human airway epithelium. For example, those multiciliated cells that you see there, also the frog epithelium has them, and it has also motile cilia on top. So when we performed our antibody staining for STXBP1, you can see that we saw all those dots in green. This is like a cell, a multiciliated cell seen from above. And all those dots correspond to the base of the cilia, if you can see from the picture next to it. So this is the base of the cilia, which is exactly the spot where this vesicle trafficking is occurring in multiciliated cells. Similarly, also the other snare proteins that we stained for, we found them in a similar location at the base of the cilia, with STX2 being also present on the ciliary axoneme. And I want to mention here that we validated these antibodies by loss of function. But of course, localization doesn't mean that this protein is actually functional at the base of the cilia. So to test for function, I performed a STXBP1 specific knockdown in the frog epithelium. And then I compared the ciliation. And you can see that the cilia are here shown in magenta. When I compared the ciliation, I found that there was a reduction after loss of STXBP1. And this reduction in ciliation was partially rescued after expression of the human wild-type STXBP1. So this indicated not only that STXBP1 is needed for ciliogenesis in motile cilia, but also that this process might be conserved in humans since we were able to rescue with the human plasmid. At this point, we wanted to investigate more about the dynamic behavior of STXBP1. So I performed a FRAP experiment in the frog epithelium after expression of the wild-type STXBP1 protein. And you can see that the fluorescence recovery after photobleaching was really fast at the base of the cilia, which indicated that STXBP1 is a highly dynamic protein. On the other hand, when I expressed 4 patient missense, pathogenic missense variants, and I looked at the fluorescence recovery, I saw that the fluorescence recovery was reduced, indicating that in the presence of these missense variants, STXBP1 is probably more bound to cellular structures and protein complexes. So far we've seen that STXBP1 localizes at the base of motile cilia that is needed for ciliogenesis, and that pathogenic missense variants alter the dynamic behavior. To test whether this data was translatable to humans, we turned our attention to measurements of nasal nitric oxide gas. Nitric oxide gas in the nasal epithelium is produced by the ciliary beating of the nasal cells. And when there is a dysfunction, a ciliary dysfunction or impairment, this leads to reduced production of nitric oxide gas. And we can measure this using a portable, non-invasive machine, which is also the diagnostic gold standard for ciliary dyskinesia. When we measured nitric oxide gas in patients and compared that to non-affected direct family members, we found that there was a reduction in the nasal nitric oxide gas, which is a sign of ciliary dysfunction. And this is in line with our hypothesis that STXBP1 has a role at the base of the cilia, and in line with the hypothesis that it might also the ciliary STXBP1 might be contributing to these pulmonary comorbidities on top of the neurological symptoms. At this point, I would like to shift gear and start talking about neurons. Maybe not everybody knows, but neurons also have cilia. In particular, they have primary cilia, which are basically this solitary structure that acts as a tiny antenna. And it's really important for neuronal function and signaling. It's so important that we have neurotransmitter receptors expressed here, like dopamine receptor 1. Somatostatin receptor 3, for example, specifically localizes to the cilium. Similarly, also key components, of signaling pathways, like the sonic hedgehog pathway, for example, are specifically localized to the primary cilium. So also these cilia have a diffusion barrier at the base that needs to be crossed by vesicle trafficking in order to deliver these neurotransmitter receptors and key signaling components. So we hypothesized that STXBP1 might have a role also here at the base of the primary cilium in neurons, and therefore that neuronal STXBP1 functions both at the synapses and at the cilia. And I want to mention here there's previous literature hinting to the fact that STXBP1 has a role outside of the synapses. I think there's going to be a talk about this later, so I'm not going to go into too much detail here. But one of the evidence that one of these papers found was that in the presence of a STXBP1 null condition, there was a strong neuronal cell death, even at immature stages, before synapses are even formed, which hints to the fact that STXBP1 might have a role outside of the synapses that is important for neuronal proliferation and differentiation. At this point, basically, I tested my hypothesis using human cell culture. So from now on, you're just going to see data on human cell cultures. And I started by testing for localization using human retinal cells. And just to give you some direction so you can orient yourself, I'm showing in magenta the ciliary axoneme, in white the basal body marker, which is located at the base of the cilium. And in green, I'm showing STXBP1 and syntaxin-2 staining. And you can clearly see from these pictures that both STXBP1 and syntaxin-2 localize at the base of the primary cilia in retinal cells. I then went on and performed another staining in human iPSC-derived neural progenitor cells. These are immature cells that don't have synapses yet. And again, also in this case, you can see that STXBP1 and syntaxin-2 localized to the base of this primary cilium. And this STXBP1 was validated by loss of function also. And finally, in the third staining, I stained for differentiated neurons. They were human iPSC-derived cortical excitatory neurons. I want to mention here that due to the differentiation process, one of my channels was already taken up, so I had to divide it into two different stainings. You can see one staining is stained for the ciliary marker in magenta, and the other one for the basal body marker. But you can still see that STXBP1 and syntaxin-2 localized at the base of the cilia. So this was really exciting for us to observe, but of course, location doesn't imply that the STXBP1 is functional here. So to test for function, we performed a CRISPR-I experiment in the neuronal progenitor cells, and we performed bulk RNA sequencing. And we found that STXBP1 was highly downregulated, indicating that this was more of a null condition than a heterozygote condition. And when we looked at all the disrupted signaling pathways, we found that one of the most disrupted, statistically significant one was the sonic hedgehog signaling pathway, which relies completely on cilia for its function. And so this was an incredible finding because it indicated that loss of STXBP1 disrupted ciliary function, which is in line with our hypothesis that neuronal STXBP1 is needed in synapses and also in cilia. And I want to conclude just by talking about sonic hedgehog. As I said already earlier, it happens completely on the cilia. It's really important for neurodevelopment because it's important for the ventral patterning and to establish excitatory versus inhibitory balance, which is important for epilepsy. And it's important also in the cortex for neuronal proliferation and differentiation. So you can imagine that for all these reasons, disrupting ciliary biology, which leads to the disruption of this pathway, is going to be really important for neurodevelopment and neuronal function. And with this, I just want to summarize the conclusions. We found STXBP1 localized at the base of the cilia. It has a role in ciliogenesis, and pathogenic variants alter patients show signs of pulmonary cilia impairment. STXBP1 also localizes at the base of the neurons, and the loss of STXBP1 disrupts the ciliary pathway. And the future direction, it would be really cool to leverage the accessibility of the nasal epithelium to develop biomarkers and monitor therapeutics. And since STXBP1 has specific binding partners at the synapses as compared to the cilia, it would be cool to leverage the specificity to parse out STXBP1's role in these two locations. And I want to conclude by thanking everybody. And sorry if I went over time. I hope it wasn't too bad. I wanted to thank people from my lab, my PI. Without her, nothing of this would be possible. People from the State Lab for collaborating, and our funding sources, and the STXBP1 Foundation for helping us fund this project, and for having me here today. Thank you so much.
Bianca, thanks so much for this great presentation. Really interesting. I have a ton of questions, but maybe the one that I want to ask is, did you look at the length of the cilia in these CRISPR-I knockdowns, or did you look at SHA receptors?
Did we get what? Sorry, I didn't understand the
Did you look at
word.
the size or the number of cilia in your CRISPR-I screen?
Oh, no, yeah. That's a really good question. It's, of course, fundamental to see that phenotype. We haven't looked at that yet because this is kind of recent data, but it's definitely one of the first things I'm going to do as soon as I go back. But yeah, that would be really cool to see if there's actually a significant ciliary phenotype. So thank you.
Yeah. And in the airway epithelium of the lungs, right? So did you look for other STXBPs? Because I know of 2 and 3 that are also expressed in lung epithelium.
We specifically focused on the STXBP1 because we were interested in this association with the neuronal and its involvement with the autism, and we were really interested in this type of protein. So we haven't looked for that, but it's actually something that would be interesting to investigate in the future because it's probably also relevant in some ways. So yeah, that's a good comment.
Great. Thanks so much.
Of course. Thank you.
Hi. I guess following up on your biomarker comment, do you expect that, let's say that we still have, we're trying to put a drug with brain delivery into people. How would that drug end up having kind of on-target activity in the nasal epithelium, and what, is there actually a path, I guess, to having the drug affect that tissue, and then for you to see an effect, I guess?
If you mean if you deliver it from basically in the brain, if there is going to be an effect also in the epithelium?
Yeah, I suppose that most of us here, at some level we're going to be trying to put it into the brain no matter what. So whether an effect in the brain can spread actually to the nasal epithelium somehow.
That's a really good question. Yeah, I'm not sure about what the answer would be. It would be really interesting to test, of course. I think the strength of the airway epithelium is also the fact that being so easily accessible, it would be really nice to keep track of these therapeutics. So if there is an effect from the brain to the nasal epithelium, that, of course, would be really convenient. I am not sure if it will spread from the brain, but it will be really interesting to see. That's a good question. Thank you.
I think that's all we have. Sorry.
Oh, I'm sorry. We can talk about this later. Sorry, I went a little over time.
Yeah,
It was my fault.